So we are delighted to have with us today Professor Domenica Bueti from CISA. Professor Bueti did her first degree in psychology at the University of Padua and then her PhD in cognitive neuroscience at the University of Rome La Spazienza. I hope I'm pronouncing that correctly. She then did postdocs at the Neuroimaging Lab in Santa Lucia Foundation in Rome and also at the Visual Cognition Lab at the Institute of Cognitive Neuroscience, UCL, London. She then became a research scientist at the Biomedical Imaging Research Center of the École Polytechnique Fédérale de Lausanne, EPFL, and at the Department of Clinical Neurosciences at the Lausanne University Hospital. Today she's a professor of cognitive neuroscience at CISA in Trieste, Italy, where she leads the Time Perception Lab. She has dozens of publications, including ones in top leading journals as Neuron, PLOS Biology, Current Biology, Journal of Neuroscience, Nature Neuroscience, and others, all concerning processing of the perception or the perception of time. She is and has been funded by multiple prestigious grants and fellowships, including an ERC Consolidator grant and 2 Horizon projects that she is the coordinator of. She is a consulting editor for Timing and Time Perception Journal and an associate editor for Frontiers in Neuroscience. She's actively engaged in communicating science to the general public and has multiple TV and radio interviews, as well as given a TEDx talk. Her research focuses on the study of magnitudes, i.e., how the brain processes space, time, and numbers. She combines neuroimaging methods such as fMRI and EEG, brain stimulation techniques as TMS, TDCS, TRNS, and simultaneous combination of 2 of them, such as TMS and EEG, seeking to understand the neural mechanisms and the cognitive architectures subserving human abilities to perceive, represent, and manipulate information about time, space, and numbers. So, Domenica, I will stop here, and I'm very happy to invite you to tell us about your exciting research. Thank you for joining us.
Thank you, Sharon. Thanks for this really kind invitation and introduction. Actually, I'm honored to be with you guys. So today I will try to give you an overview of a specific aspect of my research that, as Sharon well said, is concerns about the perception and the neural representation of milliseconds time in the human brain. But I will start by first telling you about, about time. So what is actually the time I'm interested in, right? So the time I'm interested spans from a few hundreds of milliseconds to a few seconds. And this is actually the time that is associated with your subjective experience of it, right? It's associated with the perception of time. So this is something that you spontaneously feel, for example, while you're waiting a bus, right? So in that situation, you really don't need to look at your watch to, for example, understand whether your bus is late or not, right? Because you have this sensation that the time is flowing. Same thing in front of a street light. So imagine that you are in front of this light, you arrived, you're in a rush, you're late, the light is red, but of course you don't know for how long it has been red. But of course, the longer the red light stays on, of course, the higher becomes the chance that the light will turn green. So even in that case, you are using this feeling of the flow of time to make predictions about the upcoming relevant event, which is basically the color change in the street light. So this is one aspect of it, but it's also time in this very short range is also very important for many sensory motor activities like dancing, simply moving, like playing a musical instrument. Motion is actually possible because we have a timely activations of agonist and antagonist muscles. Activity like, for example, playing tennis, like in this video where we see Roger Federer. I mean, here there are the 2 aspects that time needs to process and produce. So in one case, there is the ball, and the ball is approaching the Roger Federer. So basically your brain needs to compute, so to process the time that the ball takes to be at a reachable distance, right? But then there is another time information that the brain needs to produce, which is basically when is the right time to hit the ball. So in this case, of course, things are happening is just pure processing, and this happens without, of course, awareness, right? It's outside perception. So basically, we experience time, right, through all our senses, right? So because sensory experiences, sensory events unfolds over time, no? Like the sound of a note, or the ball that moves and approaches my body. So sensory experience is embedded in time, so this is interesting. But what is actually peculiar about time is that, of course, we don't have dedicated sensor to it, so we don't have a specific pathway for time, and this, I think, makes time peculiar as an object of research, because we have the object of our sensory experience, let's say a sound or visual image, but we also have an experience of time that is associated with the content of what I'm experiencing, right? So there are two things. And how, so the content of what I perceive and the flow of time associated with it, and how those two things interact, so is actually not clear. So basically, what is unclear is how does the perception of time interacts with the content of the sensory experience. For example, we know that the perception of time is influenced by stimulus feature, like, for example, the motion or temporal frequency, numerosity. So there are lots of sensory features that affects the perception of time, but we don't know, for example, if the coding of time is embedded in the coding of the stimulus itself. We don't know, for example, if it's sustained by the activity of the same of these neural circuits, if it's observed by similar or distinct neural mechanisms. And I'm actually here listing all these questions, not because I believe I have the answer to those questions, but because the research I'm presenting today is actually trying to have as an overarching question those big questions. And because I think that by trying to answer this question, we can gain interesting insights on the mechanisms through which the brain represents the passage of time. So I hope it's clear that my focus is the perception of stimulus duration, right? In the milliseconds and a few seconds range. And I think I can use this big questions that I just listed as also outline for my talk. So in my talk is structured in two main parts. In the first part, I will present psychophysics studies that focus on this first question. So how does the perception of time interact with the content of the sensory experience? And I will present these studies where I manipulated sensory features and see how those impact the perception of the stimulus duration. So I manipulate speed or visual contrast. And also I choose also the opposite perspective. So then I present a study where, rather than changing the features of the external stimuli, I try to change the psychophysiological state of the subject by inducing a stressor, and see how this impacts your perception of a visual stimulus and your perception of time. And then in the second part, I will move into the brain more, and I will basically try to focus on this question of whether the encoding of time is embedded within the coding of the stimulus. So, and I will specifically talk about, present you an MRI study that we conducted at 7 Tesla, where we actually tried to see how does the brain response is modulated by a stimulus that varies in two features, in duration, in display time, but also in numerosity, and how those changes affect the neural representation of time and numerosity. But let's start from the beginning. So the first set of second physics experiment. And the first is basically a study that has been conducted by a student, a PhD student in the lab, Francesca Iris Bellotti. And Francesca was interested in the relationship between the perception of stimulus duration and speed entropy. And basically, visual speed is known to affect time perception. We knew that. This is quite a solid result. And actually, so what I mean is basically what has been shown is that the faster a stimulus moves, the longer is its perceived duration, right? And these effects are can be interpreted as suggesting a sort of integration mechanism, right, that subserve duration perception, as if the greater the sensory input, the longer the perceived duration, right? But Francesca wanted actually to see whether this integration process happens, this field illusion, visual field illusion, as it is called, this effect of speed on duration, whether this effect holds when, for example, we give the subject an alternative way to solve the temporal task, so providing a sharp onset and offset. And also what she wanted to understand is whether there is any asymmetry in the speed duration effect. So we know for sure that speed affects duration judgment, but we don't know whether duration can affect speed judgments. Okay, so what she did, you will see an example of a trial in this video. So basically in every trial, you see this random dots that moves. So in every trial, we have a standard and a comparison stimulus. And what we manipulated here was both the speed of those random dots and also the display time. And those two features are manipulated orthogonally, right? What we changed was that in some blocks, we asked participants to decide which one of those two sequentially presented visual displays was displayed for longer time in case of a time task, or was which one of the two was presented, had a faster speed. Okay, so and this was one condition. So this is what you would normally do if you want to study the relationship between speed and time. But she had also an extra condition, which is the following. She, in a second condition, she had flankers to her stimuli. This is here. So she had, so the two stimuli now have still these random dots, but they are basically characterized by a very sharp brightness, increase of brightness in the onset and at the offset of the stimulus. So basically, somehow, what you would expect is that you could solve the temporal task. You could tell me how long was the stimulus displayed for by just using this sharp onset offset, and you can forget about what's going on in between those two features. And these are basically, now I'm showing you the results, and these are data for the duration task, where you see on the y-axis is the percentage of the time the comparison stimulus is judged greater than the standard stimulus, plotted against the physical actually duration of the stimulus that is presented. This is normalized to the standard stimulus, and color-coded are basically the performance of the subjects in different speed conditions. Okay, darker color means faster stimulus. So basically in the field and also in the flank condition, we see more or less the same pattern. So we see that the faster the stimulus, the longer, so we see a shift in the psychometric curve. So the longer is the perceived duration, as expected in case of field duration, but also in flank. So basically what these results seem to suggest that even though you have an alternative to solve the temporal task, you still seem to use this information that is in between the onset and the offset to tell the time of the stimulus. So sensory input matters. So this seemed to actually go in line with what I said before, seemed to suggest that perhaps you are integrating this sensory input to solve the temporal task. Concerning the symmetry of the effects, so these are the results. So here I'm showing you the results of speed, the effect of speed on, so the effect of time on speed judgments. So, and here you see that the psychometric curves are more packed, but even though they look packed, there is a statistically significant difference. So there is an effect of the duration on the speed judgments. So this means that basically, even if it in a greater extent, also duration can impact the speed judgment. So the longer the time, the faster you judge the stimuli to move. But of course, the amplitude, as I'm showing now you in this new slide, basically the magnitude of those effects is really different. So we have, of course, we have 20%, let's say, effect for time, so for speed on time judgments, but we have a much a tinier, let's say, effect of the speed, of the duration on the speed. Okay, so this first experiment goes into the direction of this integration mechanism and the importance of the sensory input for your perception of time. Then we use another, we decided to go into another visual feature, to test another visual feature, which is stimulus contrast. This is a study that was conducted by a former PhD student in the lab, Anna Piani, and in collaboration with Professor Paola Binda from University of Pisa. And basically in this experiment, the effect of, let's say, contrast, visual contrast on time perception is less defined than the effect of speed. So that's why basically Anna wanted really to better characterize this effect. And also that she wanted to check the effect of duration, of both duration and contrast changes on pupillary responses. So she also wanted to see how the perception of time affects pupil dynamics. That's why also we collaborated with Paola Binda, who is an expert in studying the effects of perception and cognition on pupil responses. Today, for sake of time, I will just present the data on the behavioral data, not the pupillary data. But okay, let's see what Anna did. So she wanted to better characterize these effects of contrast, because the previous literature was a bit controversial. So some studies show the effect, others didn't. They use a small range of contrast values. So here Anna decided to use multiple tasks. One of those tasks is a reproduction task. I'll just describe the task structure before. So basically, in each trial, a subject sees a visual stimulus that in this case is a gabor, and in this case is a patch, visual patch that has these little squares that, and here basically the square dynamically change over time. And the subject has, so the visual stimulus is displayed for different amounts of time, ranging from 400 milliseconds up to 1.2 seconds. And at the offset of the stimulus, the subject has, there is nothing on screen, but the subject has to reproduce the previously seen stimulus by basically pressing a key, keeping the key down and releasing the key, basically, once the duration reproduced by the key press matched the duration that the subject has experienced before. And what we manipulated in this case was actually the contrast level of this stimulus, right? And the contrast level here was tuned to individuals, because basically before starting the main experiment, subjects were doing, we measured the contrast detection threshold in each individuals. using a staircase procedure. The difference between those two conditions is, in this second case, the stimulus was dynamically changing over time to basically get rid of the confound that the effects of stimulus contrast on duration was simply due to the fact that there is an adaptation on the retina due to a prolonged exposure of the stimulus of the same contrast. That's why we have those two conditions. But the task structure is the same. Also, the contrast level is chosen in the same way by just using the contrast detection threshold of each individuals. But also she used a temporal discrimination task where actually subjects in each trial sees two stimuli. So in a standard and a comparison stimulus, the comparison the standard has a fixed duration, but varied in contrast, and the comparison stimulus varied in display time, in stimulus duration. And the subject task is to decide which one of the two was displayed for longer time, of the two stimuli was displayed for longer time. So we assess temporal perception in many ways then, and we try to see what's the effect of stimulus contrast. And here are the results. These are the effects of in the two reproduction tasks. What you see in the y-axis is mean reproduced duration, and the x-axis, these are the actual target duration. Color-coded is the different contrast levels. So what basically this, these are actually the raw data, and this is the estimation bias. But it's the same. These plots are telling us the same thing. So basically that the brighter, so sorry, the greater the contrast, so which is this magenta, the longer the perceived duration. Okay, so again, a similar effect to speed. And this happens also in the perceptual task, where I show you both the psychometric and the point of subjective equality here plotted. And so what you see is, again, that the higher the contrast, the greater the point of subjective equality. So in summary, basically, those two studies are telling us that the content of the sensory experience, so speed or the contrast, influences actually the perception of time. The greater the content, the longer the perceived duration. And I didn't have time to show you that, for example, we also see that there is a modulation of stimulus duration with pupillary responses. So even though there is in the reproduction phase, when there is nothing on screen, but the subject is just reproducing the stimulus duration, there is a modulation with stimulus duration of the pupillary responses. But what I think the point I want to make here is just to emphasize that really, so sensory input matters, right? The sensory input is basically the gateway, right, of our sensory experience. So, and also of time. So whatever happens at that level impact our perception of time. Then we actually decided to change strategies this time, and rather than changing the stimulus features of what we show to the subject, we decided to go around and to change the psychophysiological state of the subject by inducing a stressor in the subject. And by inducing stress, we wanted to see how stress changes your visual perception, but also your perception of stimulus duration. And what we did, this is a study that was conducted by, again, by Anna, and also in collaboration with Paola Binda and Professor Oliver Wolf, who is an expert in studies of the effect of cognition, of the studies of stress on cognition. What we did this the following. So we use a stress manipulation. We use a slightly modified version of what is called cold pressure test. So what happens is basically that we ask participants to put their hands in a water tank that is filled with ice in the stress condition, or with water at room temperature in the control condition. This actually test is well known and is well used in stress domain, and because it's known to elicit a physiological response to stress, so to elicit a change in neuroendocrine response in catecholamines, and which is usually a very fast response, and a much slower response in corticosteroids. And we basically decided to characterize over time the response to stress by measuring this physiological indices of stress, like cortisol, the salivary cortisol, but also to measure the level of catecholamines, in particular noradrenaline, by measuring also the pupil responses, okay, which is a proxy of the activation of the noradrenergic system. And we actually ask participants to perform before stressor, but also multiple times after the stressor, 2 tasks. A task that is supposed to measure low-level visual capacities, like contrast detection. In this case, subjects see a gabor that is either leftward or rightward oriented. And what we changed adaptively was the contrast level of this basically gabor. In a staircase procedure, it's 3 up, 1 down, in order to establish for each individual what is actually the minimum contrast threshold that leads to 80% accuracy responses in individuals. But we also then use this contrast threshold to also change the contrast of the gabor in a reproduction task. So we also assess temporal perception using a reproduction task, as I said, as I showed you before. Like subject sees this gabor that varies in contrast, but varies also in display time. And the subject task, after seeing this stimulus, is to reproduce by a key press the duration of the perceived stimulus. And this is actually the structure of the manipulation. So we see that we measure the pupil, the cortisol saliva, and we applied the stressor. So we measure the task and all these physiological indices before the stressor, and 3 times afterwards the stress. And to cut a pretty, long story short, in the first place, I show you that in the first place, we realized that the cortisol reactivity in our samples was pretty different. So there were subjects that that basically show a very tiny cortisol reactivity. So basically in those subjects, there is no difference between stress and control in the cortisol level, okay? Whereas there are the other extreme, subjects that show a greater response of greater cortisol reactivity induced by stress, and subjects that are in between. So they have a mild, let's say, cortical reactivity. And what is interesting is that these guys that actually show a mild cortisol reactivity are those in which we see a significant difference in their contrast detection thresholds. So basically, those are the subjects in which we see that their contrast detection thresholds becomes lower. So as if their visual system becomes just increase their acuity, right? So because the threshold is smaller in stress compared to control. And those are actually the subjects where we see a significant difference in the reproduction of time. So those are subjects in which we see that the subjects tend to, after stress, they tend to release earlier the key, as if basically the duration that they have perceived is already longer enough, and they release the key. So I think this actually results, it seemed, if you followed me, seem to be in line with what actually we showed before. So basically, if you sample more information, if your visual system increase the sampling rate, you seem to basically to be biased in your perceived duration, and duration seem to be basically longer. And this is actually congruent also with anecdotal report of subjects that reports that during stress, during a stressor event, time seemed to be frozen, right? And so a possible explanation is that perhaps it's frozen because your visual system becomes more, your sense, in this case we are of course measuring visual acuity, so your visual system becomes more more sensitive to external inputs. Okay, so this is what I just said. So lower contour detection threshold and under is associated, is paired with this under reproduction of time, as if stress improves visual sensitivity and makes duration perception longer. And this is in line with this idea that the greater the sensory input, the longer the time. And yeah, as I said before, sensory input matters. And this is actually in line with also a bunch of studies that actually show that there is a link between activity in early sensory region, in early visual regions, and duration biases, right? So there is a recent EEG studies. We actually conducted this EEG study where we show that there is a signature, there is an ERP signature, the M200, that is actually predictive of the subjective bias in time. And this seemed to be very, it's early, has this M200, so after 200 milliseconds from stimulus duration, that predicts the perceptual bias. Okay, right. Now I will move into the second part, and I will try to just dig into this more neuro aspect. So what's going on in the neural circuits when basically we manipulate time, but also stimulus features, right? And now the stimulus feature that we are exploring, it's not contrast, it's not speed, is numerosity, is the quantity of items that are displayed on the screen. And the reason we did that is because we wanted really to see how the brain response changes when we manipulated this feature, and we know that there is an effect. So that, let's say, numerosity, the number of items displayed on screen, affects duration perception, but it's also true the reverse. So these effects are very symmetric. This is a study that we published a few years ago, where we actually show, I'm sorry, I'll just go very quickly through this slide. So we show that we actually present subjects with stimuli that vary in numerosity, so the number of items or dots on screen, but they also varied in display time. And we actually ask participants to judge either the numerosity of the items or the duration of the display of these items. And we see, for example, on the left in these plots here, that if you look at the duration task, the higher is the number of dots displayed, the greater is the bias in the subjects. So the more numerous the items, the longer the time. And this is the same for the numerosity task. So the longer the display time, the more numerous are judged the stimuli on screen. Okay, so at neural level, we knew that, for example, duration, but also numerosity can be represented by tuning mechanisms and topography. What I mean is that basically that you see displayed here, these are studies that come from my lab or other labs, basically, that shows that the brain response can be to stimuli that vary in duration or numerosity, can be described can be described with a tuning function, right? So it means that there are voxels that seem to basically respond preferentially to stimuli that have a certain duration or a certain numerosity, and those tuning, let's say, those voxels that are tuned to specific duration and time are also topographically organized in the cortical surface, meaning that these clusters that are tuned to similar numerosity or similar time are also contiguous in the cortical surface. Like, you know, forming these maps of numerosity and time. And one of the goals of the study was also to try to see if I combine numerosity and time, so together in the same visual object, what is the fate of those maps? What is the fate of the representation of those features in the brain? So this is a study that has been actually coming out tomorrow in Proceedings B of the Royal Society, and it's a study that has been conducted by Gianfranco Fortunato and Irene Togoli, who are two basically postdocs in the lab. And Gianfranco and Irene wanted to understand to which extent time and numerosity recruit same different neural population, how does this magnitude integration leads to changes in brain responses, and how those map changes, just as I said a second ago. And in this case, we conducted a 7 Tesla MRI experiment. And what we did was the following. So we had subjects lie in the scanner and seeing either a condition like this. So they were seeing those bunch of dots that were displayed for different amounts of time, but the dots had exactly the same numerosity, okay? This is what we call baseline time, because we just manipulated time and we keep fixed numerosity. We also did the same for numerosity. So we had conditions in which we kept fixed the display time, but we vary just the numerosity. Or we changed the two features together in a congruent fashion, like in what you see here. So in this display, you see that the greater the number of dots on screen, the greater the display time. Okay, so we have cycles in this sense, but we also have an incongruent condition where the things is reversed. So this means that the greater the display time, the less the number of dots that are shown on screen. Okay, so we have then 4 different experimental conditions. So those two are baseline. So numerosity is fixed, we vary duration. Baseline numerosity, meaning that duration is fixed, we vary numerosity. Here is numerosity and times are changed together. Here in a congruent fashion, so the two are paired together in the same way. And this is incongruent, so one increases whereas the other decreases, okay? Here, subjects don't perform any temporal or numerosity task. Here, subjects just passively view those things on screen. There is a task like an attentional task, so subject had to detect the presence of a red dot that from time to time is displayed, just to make sure that subject is attending the stimuli. Okay, so what we use analysis-wise was a population receptive field approach. So what we show here, for example, so we basically assume that the brain response could be described by the bimodal Gaussian tuning function that is described by, is defined by 5 different parameters. 2 are the mu parameters, which basically tells you the selectivity of these tuning functions to a certain stimulus feature, so either time or numerosity. And we have 2 sigmas, which describes the spread of this tuning function, the variance of the tuning function, and a theta parameter that gives you the orientation of this bimodal tuning function, telling you basically which is the driving dimension that leads to the brain response, okay? Basically, I hope it's clear that we use a model that is sensitive to changes in principle. It's sensitive to changes of both stimulus dimension. So it's a bimodal Gaussian. And here there are examples of voxels that are fitted by our model in the 4 different conditions. Just to tell you that, I mean, the fitting worked somehow not in all the voxels, of course. And here, actually, what I'm showing in the first place is for the different experimental conditions, so baseline time, baseline numerosity, congruent and incongruent, we see the group level model fit. So here in the first place, we wanted to see the vertices where the model fit reasonably well, and to see how much overlap in these vertices there was across the different experimental conditions. And what we see is that there is not a great overlap, as expected, because has been shown previously, for example, by Harvey and Dumoulin, the numerosity and time somehow leads to distinct and only partially overlapping brain areas, right? And this overlap is greater, for example, in the baseline condition, time and numerosity, and it's stronger in occipital and in parietal cortices, and it's less, it happens less in frontal regions. But beside this, let's say, a check that we did about, okay, what are the bits of the brain that is active in our conditions, what we were interested in was just to check how the model parameters were changed with our experimental conditions. And the first thing that we checked was the aspect ratio, so which is basically the relationship between the two major axes of the receptive field, so the two sigmas, right? And this tells us what is the shape of the tuning function that best describe the brain response. So basically, if the shape is very elongated of the receptive field, it means that there is a selectivity to either one of the two stimulus dimensions. But the rounder the shape of the receptive field, the more, let's say, the brain is sensitive to changes of the two stimulus dimensions. And these are actually the results of what we see. So here we have for different ROIs, basically, that we selected based on what you saw before. So we select, so these are ROIs that goes from lower occipital, temporal occipital, parietal, more parietal, central, and frontal cortices, okay? So in those distinct ROIs, you see how the aspect ratio changes in the different experimental conditions that are color-coded. So red and blue are the two control conditions, baseline, time and numerosity, and those two are the condition where numerosity and time was changed together, congruent and incongruent. And as you expected, in baseline, the receptive field is more elongated, right? Because it means that the brain response is, so its influence is more influenced by only one stimulus feature, either time or numerosity. But in the congruent and incongruent conditions, so where the two features are changed together, we see that the receptive field is rounded. So because it means that in the same, actually, voxels, they become more, the brain response is more sensitive to changes of both features, okay? Other parameter that we check is the orientation of the receptive field, because we want to understand what is the leading stimulus dimension in, what is the dimension that drives the tuning response, the brain response. And in particular, what we actually, so what I just want to emphasize here is that orientation of the receptive field that is 90 degrees or 0 or 180 degrees means that basically the brain response is driven mainly by one stimulus dimension, whereas all orientation of the receptive field that are in between these two main orientation signifies basically that the brain response is driven by a combination of the two stimulus features. And this is actually what we see. So if you see here color-coded for the different ROIs, so here I'm showing you the baseline, okay? So time and numerosity. And in time and numerosity, indeed, the majority of the subject, all the distribution, so the subject is, the receptive field is oriented at 0 and 90 degrees, no? Has a single orientation. Whereas if you see what happens in the congruent and incongruent condition, we see a great variety of orientations in the receptive fields, meaning that actually the leading dimensions now, it's different. So it means that there are voxels that probably are, whose response is driven by either time, numerosity, or by both time and numerosity. Okay, then we look at the preferences, right? So what is really the preference of those voxels? So we look at the mu parameter. In this case, I'm showing you the preferences to duration, okay? So here is baseline. These are actually on the brain. So this is the parameter displayed on the brain. So this is the mu, the different moves color-coded according to the duration preferences. You see in red are shorter duration, in colder color are more longer duration. So this is something very similar to what Harvey and colleagues have shown in their publication in Current Biology in 2020, yes, 2020, where actually they see duration maps in a wide circuits of brain regions from occipital cortex to frontal regions. We see exactly the same here. And let's see, this is baseline. This is what happens to these maps when the time and numerosity are changed together in a congruent and incongruent fashion. I think it's pretty obvious, even by just a qualitative appreciation, that those maps change radically when the two features are combined. And we actually have a quantification of this change by measuring the distance of those vertices from one of the border. But again, for sake of brevity, I'm not going to show those results. But what you see here, it's quantified and it's true. So there is a worsening of the topographies due to the combination of time and numerosity. Same thing for numerosity, actually. So here are the baseline conditions. This is similar to what Dumoulin and Harvey have shown. Again, we see numerosity maps across these brain areas, and this is the worsening of those maps in congruent and congruent conditions. Okay, but what I'm now, this is the last really scientific bits of the talk, and then I'll just wrap up. So basically, what I want to now emphasize here is also, rather than looking at the absolute change in the preferences of those voxels, I want to show you some interesting results of how the preferences changes in the cortical hierarchy, okay? Let's see for duration. This it's actually the duration preferences in the different ROIs, so from occipital to frontal regions, in the different experimental conditions, so time baseline, congruent and incongruent. So what I think the pattern that we saw is the following. So that basically from lateral occipital to parietal occipital, basically the duration preferences have the same sort of pattern. So there is a decrease in duration preferences in all those ROIs. But from parietal occipital to frontal region, this pattern changes only for the conditions where numerosity and time are changed together. So in the congruent condition and in the congruent compared to baseline. So this means that there is really a change here in the behavior of the hierarchy, depending on whether numerosity and time are integrated or not. Same sort of pattern we see for numerosity. So numerosity is baseline, is the blue, and you see that from LO to TO, somehow the pattern is the same. So there is a decrease in the numerosity preferences. But then from PO, PC to EF, the pattern is reverse. The blue has a different pattern for the yellow. And again, so suggesting that there is something interesting happening in this moving from parietal to frontal cortices when numerosity and time are combined together. Okay, so just to summarize, we see in this last study that only partially overlapping numerosity and duration maps in this circuit of brain areas that goes from occipital to frontal. We see that the same neural units actually become sensitive to both time and numerosity when those features are combined, because we see round tuning functions, multiple orientations of theta, of the receptive fields. We see that topography degrades due to magnitude integration, and we see that the selectivity changes due to this combination of time and numerosity, especially in the transition from parietal to frontal regions. So basically, what I think this data seem to suggest is, in the first place, that perhaps it doesn't make sense to ask ourselves whether the question about numerosity and time, those two features are subserved by the same with this neural network. Probably this is not really the right question to ask. Probably because probably those two features are subserved by partially distinct pathways, right? So they have their own, let's say, neural correlates. But what I think it's more important here is to show that if those features are combined, then the brain responds. So the brain is sensitive to both those changes. And I think what also is interesting is the presence of this cortical hierarchy, no? So, and this also allow me to have a link with the previous studies that I showed you before. So in the first part of the talk, I made a lot of fuss about the role of the sensory input in the perception of time, and the fact that probably low-level sensory areas play a role. What happens there impact perception. Probably this is really the gate, the first gate of sensory information. So probably is there that time is extracted in the first place. And then there is a hierarchy in the time processing domain. There is a hierarchy that we see for many other things in the brain. So we see that probably information comes there, then is integrated in parietal cortex, where we know parietal cortex is an associative area that has, integrates multiple sensory information, is an area that integrates this information for action purposes. And then there are probably frontal regions that have a high level role because they probably read out what has been integrated in maybe parietal cortex. So this is actually my speculations that, if you want, is kind of trivial because it's a hierarchy that we know it's there for multiple sensory features. And as a last slides, just to make sense of what we know in my field, in the time perception, in the millisecond time perception domain. So what we know about time and the representation of time in the brain is that time, of course, is probably represented in a wide circuit of brain areas. It includes sensory and also motor regions. Probably when this happens, I think when we really need to ask ourselves about these different stages of the processing of time. Probably we have an encoding stage, extraction of time from sensory input. We have a decoding stage where time has to be recognized after being integrated. And probably there is, so how then probably via, again, integration, accumulation, via tuning, topography is also a possible mechanism. And of course, I presented you the maps, and the maps are shown using MRI. And because of the MRI resolution, we don't know whether really those maps that we see are really reflect the presence in the brain of neurons that are tuned to duration, or reflects more the fact that the neural circuits behave differently. The dynamics of these neural circuits is different depending on different durations. And of course, there is much more come. And I should thank you for your attention, for being patient with me, that probably talked too much. And thanks to also the people that helped me in doing this, that did the hard work, like Gianfranco, Irene, and Michele, who worked mainly on the numerosity and time experiments, Francesca Bellotti for the speed and time, and Anna Tonon for the stress and contrast experiments. And thank you again for your attention.
Domenica, thank you very much. I'd like to ask everybody to unmute yourself, and let's give a big applause to Domenica. Really, thank you for a very interesting and inspiring talk. And I'm happy to open the stage for questions, and I can start, but everybody's also welcome to ask. So I wanted to ask about the idea you were talking about, the perception or processing of the stimulus as some kind of a let's say driving force that may assist in processing or perceiving time. And I'm wondering whether, because many of the sensory, or let's say somatosensory and vision, also have a motor components. You know, the eyes move all the time, and you know, we also have motor components. And I'm wondering if you think also, you know, with the kinematics and the hands, if you think also there is some part of that, of the, let's say, efferent copy or efferent processing, let's say, that also maybe contributes to time.
Yeah, this is a very interesting question, and the quick answer is yes, sure. I think, okay, in the first place, we don't know much about, for example, faction.
Yes.
Or how, for example, you perceive time by the duration of another. This could be an interesting field to explore. But definitely, movement, it's a key component in the perception of duration. Also because I talk a lot about the incoming sensory input, but you can interpret all this as a change, right? Whatever.
Yes.
So if there is no change, it's difficult to imagine to perceive stimulus duration, right? So something that is stationary, it's, you know, that doesn't change. So it's different. And it's also interesting that most of the circuits that the brain areas that we know are engaged in temporal computations are motor circuits, right? You know, the cerebellum, the basal ganglia, the parietal cortex, the premotor areas are all real motor circuits. And actually, in a data that I don't, the area MT, I mean, one of the first study that I published in this field, there was a paper that I did in London, actually, with Vince Walsh, where we actually were stimulating the area MT, V5, while subjects were judging the duration of a stimulus. So the stimuli were, in this MT effect was present no matter the motion present in the stimuli, even with stationary stimuli. And MT is, of course, an area that cares about moving objects. And in a new experiment that we carried out in the lab, where we actually manipulated even the spatial location of the stimuli, we actually see that there is an interesting transition in area MT. So that there is also, so today I showed that the parietal cortex seemed to be special because it has this the directionality of the preference changes, changing, moving from parietal to frontal. But also MT is an area where we start seeing that there is a selectivity. This is an experiment where we try different models. It's an fMRI where we try different PRF models. And we see that, for example, whereas in V1 you have a ramping behavior. So let's say that the longer the stimulus, the greater the response in V1. In MT, you start to have a double behavior, a behavior that is ramping, but a behavior that starts to be tuned, right, to time. So as if really the transition in duration let's say, mechanism also starts from where movement is processed.
Interesting, because I also thought really to ask you about if you have looked in the results you were showing, really going from, let's say, lateral or parietal to frontal, and I was wondering if you, if you sampled, even if you didn't have there if you sampled V1, because that would be, the first step and the very basic sensory, empty, of course, for motion. But I was wondering about that, and I will continue as. One more question is, I want to ask if you think that, so one thing that came to my mind when you say that you need changes in order to sense time. So there's this experiment where people or a person close themselves in some, white room for a few days, and they are, and they lose the perception of time. They know they're supposed to be there for, let's say, 3 days, but they are completely all the sense of time, because there's no change. They're just locked with themselves. So it really affects their perception of time, which maybe, I'll look it up and I'll send it to you.
Thank you. It's a very interesting suggestion. And this reminds me also what also in the electrophysiology fields of people that study, that explore time perception in electrophysiology, so that the focus has switched from just looking at the neurons and see how individual neurons or populational neurons, and see this ramping behavior, right, as a proxy of time representation. Then they move and see, for example, these Jazairi studies, where he shows that basically it doesn't matter the amount of ramping in the neuron. What matters, it looks like, it's the change, how much change there is in the network dynamics. They seem to be linked to the duration, the physical duration of the stimuli. So the faster is this dynamic of the neural network, the shorter is the duration. The slower the dynamic of the system, the longer the time.
Okay,
that's an interesting parallel, an interesting
yeah, because I wanted to ask if there's any measure in brain activity that you can say that some accumulation of activity would predict time, and you're saying that it's the accumulation of change in a way predicts so it's some relative to how the network usually works. Great. Are there additional questions? Well, okay, so I did have some additional question, but I think I will continue with this with you offline, Domenica. Later on. I've got a couple of questions about your studies. Yeah, definitely. So I again want to thank you a lot for joining us.
You're welcome.
For this really interesting talk. And I'm not sure about next week. If we do or don't have a talk, we'll keep you posted, because anyway, we had a cancellation, a last minute cancel, not a week notice, but so thank you very much for this great talk. And
yeah, we keep in touch.
Yes, we keep in touch. I want
to actually advertise that I'm looking for PhD students. So we have an open call that closes in August. So if you want to join SISTA, so we are looking for PhD students in beautiful Trieste.
Yeah, lovely.
At the border with Slovenia. So if just people don't know, so it's 1.5 hours away from Venice. And
wonderful.
Yeah.
So yeah, we will. If you post it, I will. I will. Yeah, lovely.
Thank you very much for this.
Thank you very much, Domenica.
Bye.
Thank you.
Thank
Ciao.
you. Ciao.